at the structure of nucleic acids. In general, we talked of the basic units of nucleic acids being nucleotides. And we talked about nucleotides uh, being composed of a pentose, that is a 5-carbon sugar, with a nitrogenous base on the carbon number 1 of the pentose, and the phosphate group on the carbon number 5. And we went ahead and talked about the type of bonds that exist between the nucleotides to form a nucleic acid strand. So we talked about the phosphodiester bond. And we also talked about the secondary structure of DNA, that is having a double helix structure, where we talked about the base pairing that a purine will always pair with a pyrimidine, such that adenine in DNA will always bind to thymine and guanine to cytosine. And the bond that exists between these bases were hydrogen bonds. And we said in RNA, in place of thymine, you have uracil, so that if you are having any base pairing in RNA, then it's going to be adenine and uracil. That was the difference between DNA and RNA molecules in terms of the bases. We looked at the difference between the sugars that are present in DNA and RNA. That in RNA it was ribose, in DNA it was deoxyribose. And we saw the difference in the structures of these two sugars to be on carbon number 2, where in RNA you have on carbon number 2 of the pentose hydroxyl group, that is in ribose. But in deoxyribose, which is what you find in DNA, on carbon number 2 of the sugar, you don't have a hydroxyl group, you have 2 hydrogens on that carbon. So basically that was what we did in our previous lecture. Today we want to continue and look at the structure of RNA molecules. If you remember, we ended with the secondary structure of DNA. So we are continuing today with the structure of RNA molecules, and then of course we'll look at we'll look briefly at a higher a higher level of organization of the structure of DNA along the line. The structure of DNA does not end just at the secondary structure, but looking at the way the course is structured, it's okay if we just end it at secondary structure for now. As we go ahead, we will briefly see a higher level of organization. You will see the let's say tertiary structure, quaternary structure of DNA. All right, so we can go into today's lecture looking at the primary structure of RNA. So just as we said previously, that the primary structure of DNA was the sequence of nucleotides in the nucleic acid strand. It is the same for RNA too. Remember, we compared that to, say, the primary structure of a protein is the sequence of amino acids that are bonded together to give you a peptide. Similarly, in nucleic acids, the primary structure is the sequence of nucleotides that bind together to give you the nucleic acid strand. Now, we said earlier that RNA molecules are single-stranded. They are not double-stranded like you find in DNA. Then the question arises, so how do we come about talking about base pairing in RNA, saying that in place of thymine you have uracil, in that uh, pairing of bases in RNA molecules? Now, you can have what we refer to as a secondary structure of RNA molecules, just as we have in DNA, except that in RNA molecules, it is not two separate strands that are bonded together. They are bonded in a manner such that there is that base pairing between two separate strands. But you can have what, for instance, we refer to as a hairpin structure of RNA. Now, what does that mean? This is just a single strand. This is not a double strand that you are having what we refer to as the stem region. So if you have an RNA molecule that folds such that at a certain point you are having complementary bases that are close to each other such that you can have hydrogen bonding between these bases, then you have what you refer to as a stem region of that RNA molecule. Now, when you look at this structure, it looks just like the the hairpin that the ladies use to hold their hair. That's why it's being called a hairpin structure. Now you can have also what is being referred to as a stem loop. Really, what you see here is similar to what you are seeing here, except that because you have a stem region, meaning that yes, you have an RNA molecule which folds, and at a certain point, you are having complementary bases close to each other such that you have hydrogen bonding existing between them. But then somewhere also along the line, you don't have that complementarity, and so there is no bonding. So you can have what is being referred to as a bulge. So you can have some portion of the RNA molecule that is having the stem region where there is the hydrogen bonding between complementary bases. And you can have some other areas that you don't have such complementarity between bases. And so then you have what is being referred to as a bulge. Now, such a bulge can occur in different ways. So here, when you look at it clearly, you see that these are three bases that have no complementarity bulge outside. But you can have what is referred to as a single base bulge. So it's just one base that has bulged out, a single base bulge. Or you can have what is referred to as a mismatch. So for instance, if you have adenine here and if you have adenine on the left hand side and you have adenine on the right hand side, of course the two of them, they do not they have complementarity. Or if you have adenine on left hand side, you have guanine on the right hand side, both of them are purines. They are not going to have such complementarity. They will not have any hydrogen bonding between them. Or you have adenine and then you have cytosine. So even though you have bases that are opposite each other, they are not uh, they are not complements of each other, and so there will not be any hydrogen bonding between them, and they bulge out. That is what is being referred to as a mismatch. It looks it's just like what you are seeing here, single baseball, but except that here it is occurring at only one side. Here it's occurring at both sides. There's a mismatch. Means that here there is actually no base opposite it. It has actually just bulged out and there's no base opposite it. Here you see there's a base, that you have two bases that are opposite each other, but they are not complements of each other, and so you see that you don't have such hydrogen bonding between them. You can also have, so in fact, what you refer to as a symmetric internal loop. So here you have bulging out on both sides, and you see that 3 bases are on the left-hand side and 3 bases are on the right-hand side. So the number of bases that are mismatching on both sides are equal. And so you have what is being referred to as a symmetric internal loop. Obviously, you can also say this is also symmetric. The mismatch is symmetric, except that it is specifically to do with single bases on both sides. That is also symmetric. If you have on one side more bases that are bulged out than the other side. So here you have 3 and here you have 1. It can happen that you have 3 here, you have 2 opposite it. Or you have 3, you have 4 opposite it. You have 2, you have 1 opposite it. All that is asymmetric because you have unequal number of bases opposite each other that are bulging out. And so you can have these structures as the secondary structure of RNA molecules. Now, a common RNA molecule that has a secondary structure is the transfer RNA. So you can note it down when after the lecture you can actually go and look at the structure of the transfer RNA, and you will see clearly that yes, you have portions of the transfer RNA having this secondary structure. So actually, yes, you have portions of it having secondary structure, but you don't have only one secondary structure in it. You have more secondary structure inside there. So that will take us to what we refer to as the tertiary structure. Now, the tertiary structure only means that you have an RNA molecule that is having more than one secondary structure in it. So as I was saying, that when you take the transfer RNA, you look at the structure, you will see that you have secondary structures in it. And those secondary structures is more than one. So because they are more than one, you refer to the structure of the transfer RNA as a tertiary structure. So for instance, what you are seeing here being referred to as a pseudoknot. You see, this is one stem region. So it is just one RNA molecule which has folded twice such that you are having 2 stem regions. And one stem region is forms a secondary structure. The second one forms another secondary structure. And so because you have 2 secondary structures in it, the entire structure is a tertiary structure. That's all what it means. So if you take any RNA molecule and you are having more than one secondary structure in it, then the structure of the entire molecule is a tertiary structure. And you see that this same pseudoknot, which is represented here, is given much more, in detail, such that you can see that where the stem region is, you are seeing that there is that complementarity between bases. So you see guanine, is having cytosine opposite it, uracil having adenine opposite it, cytosine, guanine, cytosine, guanine, cytosine, guanine, cytosine. So you see that complementarity between the bases close to each other such that there will be hydrogen bonding between them to form the stem region. Intentionally, these are written in green to correspond to the green stem region that is showed in the structure of the pseudoknot. Similarly, on the blue side is also written much more into detail so that you can see that yes, you have bases that are complements of each other, and so you have such hydrogen bonding between them. Then you look at the red regions which are not having any complementarity, and you see that they are not having any sort of hydrogen bonding with bases that are opposite them. It is the same structure which is drawn also here, so that here you are seeing that you have some sort of a backbone with a sugar showing in it, and then there is a nitrogen base, so that you just to represent that yes, you are having the bases in there that are having such hydrogen bonding interaction between them. So it is the same structure which is being represented in three different ways for you to look at. Now, I mentioned that a common RNA molecule that you will see secondary structure in it was transfer RNA, and you have more than one in it. And so the complete structure of the transfer RNA is a tertiary structure. But we have different RNA molecules that have such tertiary structure. Many of them are what we refer to as ribozymes. So ribozymes are catalytic RNAs. In other words, these are RNAs that act as enzymes. So they go into catalyzing some chemical reactions. And most of the times, the type of chemical reactions that they are involved in is, is to do with processing of the RNA molecule itself. And mostly it's about cutting off of, or let's say, restriction of RNA molecules. It can be in the process of, say, recycling of an RNA molecule. That is, you have the RNA molecule broken into its components, first the nucleotides, and then later it can go into, those nucleotides can go into the synthesis of other nucleotides, or maybe even broken down, the sugar separate, the nitrogenous base separate, and all of that can go back into the synthesis of new nucleic acids altogether. If it breaks down all the way to even the nitrogenous bases breaking, of course, the nitrogen in it will go even into the synthesis of proteins, because we know proteins have some the amine group where nitrogen is. So when you look at the biosynthesis processing in living organisms, it's a very complex process, such that you can have any of these molecules breaking down, and you have some of these elements going back into recycling. And you find ribozymes acting in the case of breaking down RNA molecules. So you have some which you see, this is referred to as a lead zyme. These are some RNA molecule. You see, it's just one strand, but then you have some bases that are close to each other that you see that there is some form of interaction between these bases. This is looking much like a hammer, and so it's being referred to as a hammerhead ribozyme. You don't bother yourself so much about these names. These are just for you to see that, yes, you can have RNA molecules complex, to the point that, yes, you refer to such structures as tertiary structure, because you are having base pairing at different regions, and a region that is a stem region where you are having a base pairing. One of them, if you have only one, then you have a secondary structure. But if you have more than one, you have a tertiary structure. So this is not for you to be looking and say, oh, do I have to know how to draw these? And no, it's just for you to see that you can have such a complex structure of RNAs, even though they are single strands. You can have base pairing occurring in them such that they form secondary and tertiary structures. Then, of course, you can even have quaternary structure. So I mentioned to you that, yes, at a higher level of organization, you can have even a quaternary structure of DNA. Now, when you talk of quaternary structure of RNA, it only means that you are having usually it's a complex of RNA and proteins, or even if it is only RNA molecules that you are referring to, it only means that you are having RNA molecules that are having tertiary structures in them. But these RNA molecules form complexes together such that the complex acts as one. So, for instance, when you take proteins, you talk of the quaternary structure of proteins. And one common example of quaternary structure of protein is that of the hemoglobin, such that the hemoglobin is made up of 4 separate subunits. These are proteins on themselves. These are peptides on themselves, but they act as one unit. The 4 subunits come together and act as one unit. And so that is a quaternary structure, because in each of the proteins, you have, say, secondary and tertiary structures, but all of them come together to act as one. It is similar thing with RNA molecules, such that you have RNA molecules, let's say 2 or 3 of them, and each of them is having tertiary structure in them. And the combination of these RNA molecules, each of them having tertiary structures in them, but they are forming a complex to act as one. That gives you what we refer to as the quaternary structure of the RNA molecule. Now, before we continue, we have just dealt with the structure of RNA molecules. So we'll say that we have dealt with the structure of DNA, dealt with the structure of RNA molecules. And I told you from the beginning that our studies in biochemistry, in this course is such that we take some biomolecule and we try to look at what it is made up of. That is the structure of it. After we look at the structure of it, then we want to look at the function. What does the molecule do? So right now we are going into the function of nucleic acids. But before we go into this function of nucleic acids, I think that it will be easier if you look at this function in a 3D, let's say in form of a video, before we start to look at this on with a screen. If you want to just be looking at it in the screen, it may be difficult to understand. So I will show some animations first of all for you to see, and then from there, when we look at what is on the slides, you'll be able to have a better understanding. And especially when you go to study yourself, prepare yourselves and all of that, you have a better understanding of what we are talking about. So let's watch some few animations.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right, so that was the process of replication. We go ahead and look at what we refer to as transcription, DNA transcription.
In order for our bodies to function,
let's watch this transcription first of all.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the Tata box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the Tata box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach. These complexes prepare the DNA for the successful binding of RNA polymerase. Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now, energy must be added to the system for transcription to begin. This energy is provided by the reduction of ATP into ADP and PI. RNA polymerase then synthesizes an RNA template from the strand of DNA. Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates and the newly formed strand of RNA is released.
Okay, so that was the process of transcription. So we bring all of these together and look at the process of replication, transcription, and even the process of translation, that is protein synthesis.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions Instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in three stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliciosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down one codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum,
Okay, so
modified as it passes through the Golgi.
okay, so that was the process of replication, transcription, and translation that we looked at. And these three processes are the functions of nucleic acids. That is what nucleic acids do. And so we say that the nucleic acids store the genetic information and which is transferred from parents to offsprings. And what are these codes for? So the final product that comes from these codes are proteins, as you see from the video. And this is what we refer to as the central dogma of molecular biology, which simply is to say that the code which is in DNA is first copied onto a messenger RNA by the process of transcription. And the messenger RNA, after having that message copied onto it in the nucleus, then moves into the cytoplasm, where you now have the process of translation, which is also referred to as gene expression. That is to say, as the word suggests, translation, it means the code is being translated. What is the code for? The code is for the synthesis of a specific protein, and that protein is now synthesized. Now, in the last video, you saw that it was Taylor-Towers, the synthesis of a protein that becomes an enzyme. Right from the beginning, it was related to digestive enzymes. But of course, the process of translation will produce proteins that may have different functions. So it will produce different proteins. Yes, some proteins will become enzymes, but not all proteins become enzymes. Some of the proteins could become some neurotransmitters. Some of them could become hormones. Some of the proteins can become just structural proteins, like proteins in skeletal muscles. So for different purposes, you have these proteins being synthesized. So you see that in the first video you watched, that was specifically on the process of replication. And that was about opening up of the DNA molecule, because it's a double strand. The hydrogen bonding between the nitrogenous bases, the complementary bases, will first of all have to be broken, and the molecule had to be opened up, be exposed before the information, the code, can be copied onto a messenger RNA. And that is the process of replication. Now you see that that first video was Taylor-Towers having the synthesis of a new DNA molecule from an old one, which was referred to as a process, a semi-conservative process, because in the end you are going to have two DNA molecules that are made up of one old strand and one new strand. So that will be occurring in, say, the process of cell division, where you are having the DNA molecules, having, being replicated into two separate uh, molecules that will go, one of them will remain in the parent cell and the other, will go into the new daughter cell that is going to, be reproduced from that uh, parent cell. But if you look at it in the second and the third video, you realize that replication, when it occurs, you have the opening of the DNA molecule for the bases, the codes to be exposed. But you don't have new DNA molecules being produced out of it. What you have is a messenger RNA. It's just to copy the information from the DNA molecule onto a messenger RNA. And then the messenger RNA dissociates and the molecule closes back. The DNA molecule closes back. So even though the process of replication as it occurs in say cell division, where you are producing two separate DNA molecules, is the same as the process which is occurring in the protein synthesis, gene expression. You find that in the process of gene expression, that is a synthesis, you are not having two separate DNA molecules being produced from it. You only copy the information. The information is only copied onto a messenger RNA, which later comes out through the nuclear pore from the nucleus into the cytoplasm for the process of translation to occur, and then there is that synthesis of a protein out of it. So generally, we have just gone through that full process of what the DNA is for. The function of the DNA is as it is having that genetic code. What is the genetic code for? The genetic code is for the synthesis of proteins. And these proteins have different functions as they are produced. They go, they have different functions. They can be enzymes, they can be neurotransmitters, structural proteins, different things, hormones in the body. Now we can look at these processes much more into detail. So if we are looking at the very first process, which is the process of replication, for instance, you saw in the video what was referred to as a replication fork. That is when you have the opening up of the DNA molecule and you have different protein complexes that are involved in the process of the synthesis of the new DNA strand, when you are seeing the base pairing occurring. And on the leading strand, you find that the new DNA strand that is in orange is being formed in a continuous manner from the 5' end to the 3' end direction. And you saw that various proteins were involved in the process. So for instance, in this diagram, you are seeing what is being referred to as a DNA gyrase. And clearly, when you look at it with an arrow on it, it's telling you that what this protein is doing is just to unwind. Remember, the DNA molecule is a double helix structure. The secondary structure is a double helix structure. So clearly, it's just like as if you are using some. Well, this either you say it's a screwdriver to say or unlock a screw or to unscrew. something, or maybe you are using what you call a spanner to unscrew a bolt, for instance, something like that. So DNA gyrase is having such a function in this process. Then you also see things like okay, this strand is labeled as the leading strand. It means it is on that strand that you find that the synthesis of the new strand is in a continuous manner from the 5' end to the 3' end. You also see things like the the beta subunit sliding clamp. Clearly, it just shows that these are proteins that are also, all these proteins are involved in stabilizing the replication fork 4, the process of replication to, occur. So you have something like a stable proteins, stable binding proteins, SSB. You have dimeric replicative DNA polymerase. So clearly these are DNA polymerase. So that is responsible for bringing the nucleotides together to form a polymer. So the name will tell you that this is for a polymerization reaction, meaning that it is responsible for bringing the nucleotides together, binding to form the strand. Here it is, if you say dimeric, dimeric only means that it is made up of two separate subunits. So you can see the first subunit here and the second subunit down there. That's why it's being referred to as a dimeric. Replicative is it is acting in the process of replication. So all of these are just terms to refer to for you, that when you see the name of the enzyme, then you will know specifically what that enzyme is doing, or call it a protein, what it is doing. Dimeric because it is made up of two subunits. The process in which it is involved is replication, and so it's a replicative. And the specific thing it's doing is to bring nucleotides together in a if any of you is calling, maybe the person should stop the call so that it will stop distracting us. Okay, so you see on the lagging strand, that is the strand on which you have the synthesis of the new strand in smaller chunks. That you saw in the video, and these smaller chunks are referred to as Okazaki fragments. So you take note of that smaller chunks. So, and also take note of the fact that the synthesis always is occurring in the direction of 5' end to the 3' end. So you can see here, this is the 5' end going towards the 3' end. Here too, because this strand, the old strand, this is 5' end. And so the synthesis of the new one is starting from its 5' end. Again, just the same direction to the 3' and 5' end to the 3' end. But this time around, it is in smaller chunks. It is in chunks, being referred to as Okazaki fragments. Here too, you have DNA polymerase, and then you are also here the DNA ligase, which you saw in the video, responsible for binding the various DNA molecules, the various nucleotides, making sure that there is that seal, the bonding between them. That is basically concerning the formation of the phosphodiester bonds between the nucleotides. You also see the green portion, what you saw in the video, this being referred to as the primer. So first of all, you have the RNA molecules. Remember that you see, it's just like I said from the beginning, that it is the same process of replication which is occurring in the cell division, where you are having the production of two separate DNAs to go into two separate cells. It is that same process which you are seeing also occurring in the process of the protein synthesis, where first of all you have that process of transcription, which is the copying of the RNA molecule. It's the same thing. So you see that in the beginning, first of all, there is an RNA primer which comes in. So all what it means is that in place of thymine, the uracil is what is binding in that primer. That is why in the end, that primer is removed. So that now you now have DNA nucleotides coming in. So if you remember, you know that the RNA molecule is having a ribose in it, but the DNA molecule is having deoxyribose. So what it means is that in these primers, you are going to be having the ribose sugar, and the base that will be in place of thymine is going to be uracil. So in the end, if you are going to have two separate DNA molecules, these primers will have to be removed. So the primers will be removed and then replaced with deoxyribonucleotides. So that's the difference between the primer and the nucleotides that you are supposed to be, that you find in DNA. The primer will be having ribose, let's say ribonucleotides, because the sugar in it is going to be ribose. And in place of thymine, you are going to be having uracil. So they are removed so that deoxyribonucleotides will now be replacing them. So that is about the exchange of the primer for the appropriate nucleotides to be bound onto it. So when I'll actually share the videos to which I'll give the videos to you so that you can watch it as many times. Sometimes when you watch some of these things just once, you may not be able to understand fully, but if you watch it multiple times, you'll get a better meaning out of it. So I'll share the videos with you. You look at it, and then you also listen to the explanations and also the slides, so you have a better understanding of this process. All right, so we are only looking much more into detail of this process, so that after replication, you have the process of transcription. And here too, just as you saw in the video, you are seeing this is a messenger RNA molecule, which is being transcribed, or say copied, from a DNA molecule. So it's a portion that is a gene coding for a specific protein being copied as a messenger RNA. Now, you saw that in that process, you saw it is divided into 3 different stages, which was initiation, elongation, and termination. So you pay attention to that the process of transcription is in 3 stages: initiation, elongation, and termination. Now, you may also pay attention to the enzyme RNA polymerase. So you heard of DNA polymerase, and I was telling you, when you look at the name of the enzyme, you will know what it does. So that when you see DNA polymerase, you know that that enzyme is responsible for bringing nucleotides together, binding them together to form a polymer, which is DNA. in this case, this enzyme will be bringing ribonucleotides together to form the RNA molecule. It's a polymerization reaction. So the final product, which is an RNA, is a polymer. It's a polymer of what? It's a polymer of nucleotides, ribonucleotides, which means the ribonucleotides are the monomers that come together, they bind together, that phosphodiester bond to give you an RNA molecule. Now we can move on and look at this process of initiation. So what we are doing now is we said that the transcription is in 3 stages: initiation, elongation, and termination. So let's look at that process of initiation, elongation, and termination, much more. Let's zoom into those stages. So again, in the video, you saw that there was a region of the gene being referred to as a promoter region. And such promoter regions are of some specific nucleotide sequences that are recognizable. So we are being referred to as recognition sites. So it means that the transfer RNA that is bringing an amino acid, the ribosomal subunits that come to bind to form the complex, they recognize these recognition sites. So they come to bind to this specific site. Then there is the portion which is the gene, the code itself that should be copied and then be translated into the protein. That is being referred to as the transcribed region. Then you have so to this promoter region, you now see that you have the enzyme RNA polymerase binding onto it. So the RNA molecule binds onto the DNA molecule. Then you have that opening up of the strand. So that, of course, you refer to as replication, because these two, the strand has opened up, exposing the codes. This is now exposing these nucleotides so that they can be copied. So you see RNA polymerase binds to the promoter region, specifically because of these recognition sites, and causes the opening up. So that will be an initiation of the process of transcription. Now, when that happens, you now see that there is that synthesis of the RNA molecule. So the messenger RNA is now being transcribed. Again, you see, from the 5' end to the 3' end. So that process goes on. So you see the RNA polymerase is responsible for catalyzing that process. The strand, the DNA strand, is being copied as a messenger RNA. So the messenger RNA is the orange line. And again, take note, the synthesis is in the direction 5' end to 3' end. Now, after you have that copying in that process of elongation, so you see clearly, it's just the word even tells you elongation. So one nucleotide after the other is coming to join onto it all the way, as you saw in the video, to give you the full RNA strand. Now the RNA strand, at the point when it reaches the end, which was the terminator region, then you see the RNA molecule dissociates, the whole complex of the RNA polymerase dissociates from the DNA molecule. The DNA molecule zips back. The messenger RNA dissociates, and then it can now move from the nucleus through the nuclear pore into the cytoplasm. Now you notice that there is something interesting here. At the end, this termination, you see the process which is occurring, causing this RNA molecule to dissociate from the terminator, is forming a secondary structure. So this should tell you that, okay, even the hairpin structure has a function in the process of transcription. So this structure is what we saw, the first secondary structure of RNA, which is the hairpin structure. So it's almost like the RNA molecule is forming a knot at the end, zipping together, forming that secondary structure, and thus dissociates away from the DNA molecule. That is the template that the RNA molecule is being copied from. So you can keep that as one of the functions of the hairpin structure. Or even generally, you can say the secondary structure has this function of termination in the process of transcription. Now, you also saw in the video that there was reference to the genetic codes. So I know in your first year, you had done something on genetic codes as well. So you can revise on these things. But perhaps if you didn't pay much attention to certain things, so you don't necessarily have to, kind of say that you want to keep all of these codes in your mind. There's no need. At any point in time, it's a reference. You can always refer to these codes. But you should keep in mind at least what we refer to as the start codon, which is adenosine. So then you have uridine, you have guanosine. Of course, all of these are monophosphates. So AU gene, this code, codes for methionine. That is one of the amino acids which you learned in your first year. Now, what it means is that each time in the process of protein synthesis, the first amino acid that starts that process is methionine, because the start codon codes for methionine. So you keep that in mind. Now, I think this one should be easy to remember. The start codon. If you forget at all, if you just remember the month August, the short form of August, AUG, you should remember it. So the start codon, AUG, codes for methionine. And that is telling you that the first amino acid to start the process of translation, that is protein synthesis, also referred to as gene expression, is always methionine. Then you also keep this, you also pay attention to these three codons that are also, they are responsible for the termination of the process of translation. We refer to them as stop codons. These are UAA, UAG, UGA. This one too, it shouldn't be that difficult for you. If you look at this, UAG, UGA. I mean, the same nucleotides that are in there. You only swap this. They are always starting with uridine monophosphate, uridine monophosphate. So the only thing you see is that for these and these, yes, the adenosine monophosphate comes before the guanosine monophosphate. But here, you see, it just swaps. So you can remember this easily. UAG, UGA. and then this is UAA. So these three stop codons, that too you should keep in mind to know that these three codons, they don't code for any amino acid. That's the reason why when it gets to that, then there is the termination of the process. So if you are having 64 codons in all of these genetic coding, and you are having 3 that are not coding for any amino acid at all, it means that it is only 61. So only 61 codons code for different amino acids. And you can see that they are labeled to show that. So for instance, these codons code for phenylalanine. These ones will code for leucine. These will also code for leucine. These will code for isoleucine. These will code for valine. These will code for serine. This will code for proline. This will code for threonine. This will code for alanine. And these will code for tyrosine. These will code for histidine. These will code for glutamine. This will code for asparagine. This will code for lysine. This aspartic acid or aspartate. This glutamate or glutamic acid. This will code for cysteine. This will code for tryptophan. This arginine. This serine. These two arginine. And then the smallest amino acid, glycine, you find it also being coded for over here. So, like I said, it's not that you should be keeping all these codes in your head that this codes for this particular amino acid, because there's no need doing that. But at least when you see the code, you should be able to tell what it means that these codons are coding for this specific amino acid. All right. Then the protein synthesis process itself. The protein synthesis process itself. You saw in the video that the complex that is formed by this ribosomal unit on the messenger RNA, that messenger RNA is having 3 different regions, which were referred to as the E, the P, and the A. Now, you see that here, you see the first amino acid which is there is methionine. And then you see that this is a transfer RNA bringing another amino acid. This is phenylalanine coming to the A site. Now, what are these EPA sites? What do they mean per se? So, I mean, E stands for exit. So you saw that in the video. That is where the transfer RNA that gives out the amino acid that it brings to bind onto the amino acid that comes after it. That's where it leaves. So the transfer RNA leaves the complex from this site. That's why it's being referred to as E site. It's an exit site. So the E is coming from the word exit. Now the P site, as you can see clearly, this is where you are seeing that peptide bonds are being formed between the amino acids. And so what you are having, the process you are having in the P site is forming a peptidyl. So the P stands for peptidyl. You can say peptides are being formed. A peptide is being formed. I mean, this is showing methionine, tryptophan is you can say this is a dipeptide. So peptides are being formed here, and so this portion, this site is being referred to as a P site. The P standing for a peptidyl site. A site. So this is where the amino acid comes into the complex. So the A stands for amino acid. because it's not just an amino acid that is coming in, but the amino acid is bound onto a transfer RNA. So it's being referred to as, that's an amino acid. So this is the amino acid site where the transfer RNA brings the next amino acid to bind to the ones that are already there, to continue the process of the joining, the formation of the peptide bonds between the amino acids to form a peptide that can become a big protein in the end. Right. Now you see, so it's just the process of initiation, elongation, termination, which is just being shown again, just being shown different steps, so that yes, in the beginning, the first amino acid which is coming in the initiator transfer RNA is carrying methionine. So that is the start codon carrying the methionine. Actually, this is a mistake over. Well, okay, it's not a mistake. This is the anticodon because it's a transfer RNA. It's a transfer RNA that is having that anticodon, and the codon itself on the messenger RNA. You see, this is AUG. That's a start codon. So you see the A is complementary to the uracil U. The U here is also complementary to the A. The C complementary to the G. So you see that the transfer RNA is actually having that three bases that are complementary to what is on the start codon. And that is referred to as an anticodon on the transfer RNA. So clearly you see that this is the first one. The first amino acid is methionine for that initiator transfer RNA, starting the process of translation or gene expression or protein synthesis. They mean the same thing. Now, after, so all that, it comes in, it comes with the large ribosomal unit binds to the large ribosomal unit binds to the promoter region, and then you see that there is that shift all the way until you have these complementary bases bonded together. And you have so that's the first of all, the small ribosomal unit comes in first before the large ribosomal unit then comes to form the whole complex, where you now have the exit site, the peptidyl site, and the amino acid site now together. So you now have the whole of the initiation complex being formed. So the complex is now ready for the process of translation, which is gene expression or the synthesis of protein to start. Now, so what we saw there, what we saw earlier, see, I was talking about the first amino acid which you find in the synthesis of proteins is methionine, but that is in eukaryotes. In bacteria is a little different. So let me say in prokaryotes is a little different. The amino acid which is the starting of the translation process is formyl methionine. So it only means that the methionine is bonded onto a formyl, call it a residue, a formyl residue. I mean, it's coming from formic acid. So it's just that the type of methionine which you find in bacteria in that process of initiation is slightly different from what you see in eukaryotes. And so in prokaryotes, you see, if you see F-met, it only means formyl methionine. Now, another thing is that, so okay, let me say that, in fact, because you find this difference of methionine, the formyl methionine in bacteria with what you find in eukaryotes. So really, you can use the amount of formyl methionine which is found in the body. Let's say if you there is some formyl methionine test which is run to test for infection, so that if formyl methionine is detected in a biological sample, blood, then it can be confirmed that there is some infection, because it's supposed to be in bacteria. We don't have that. What we have is methionine. So if you see formyl methionine, it means there is some form of, infection. So you have such formyl methionine test which is run to actually diagnose infections. Now you see here this is the small ribosomal unit, and later the large ribosomal unit comes to form the pull initiation complex. Now, we refer to specific portions that the RNA polymerase, the complexes like this initiation complex, the small ribosomal unit, the large ribosomal unit, the code on the messenger RNA, which is recognized, that you find this complex binding to initiate the process of translation as a promoter region. In bacteria, we refer to it as Shine-Dalgano sequence. So you have this guanosine, adenosine, guanosine, GG, AGG, the Shine-Dalgano sequence. So in bacteria, that promoter region for the initiation of translation is referred to as the Shine-Dalgano sequence. Now we'll look, as we continue, we'll look at what we refer to as operons, the gene in bacteria. You know that in bacteria, prokaryotes, they don't have the nucleus, but they have genetic material. The DNA which is present in bacteria is what we refer to as plasmids. They are circular, and they are found directly in the cytoplasm, and we refer to them as operon. Actually, we'll come to what we refer to as the operon concept, and we'll see what we are talking about. So we'll talk more on operons. The process continues, elongation. So the first, it just shows that you have this transfer RNA bringing a certain amino acid onto the A site. This is the first in the P site already, and of course that is going to be, say, methionine, and then you have a second one which is brought. So this is the second one which is coming to the A site. After it comes, you will see the first one now goes onto the second one, forming a peptide bond between them. So you now have a dipeptide. Then there is that what you refer to as translocation. So there is the movement of the complex to the right. So there is that shift, so that you now have this transfer RNA, which just gives out the amino acid to bind onto the first, is now empty. And so when there is the shift, this transfer RNA now goes into the E site, the exit site. And from the exit site, you see it leaving the complex to go and bring another amino acid. And the process is repeated over and over. Transfer RNA is bringing amino acids, and the process continues on and on and on. Now you see here that the methionine forms the N-terminus of the polypeptide. Now, all what it means is that if you take the amino acid methionine, so if you see, if this is the methionine which is there for the first time, and then there is a new the second amino acid which is coming in, you see that it is the first amino acid that moves to come and bind onto it. Now, what it means is that when that process continues, the first amino acid, which is methionine, will remain the first amino acid at the beginning of the peptide chain which is formed. And the end of it there is the amine group, the N terminus. So you have to go back and revise the structure of amino acids. I mean, take a jotter, draw the structure of methionine, or draw the structure of any amino acid and look at the amine group, the carboxylic acid group, the alpha carbon, the side chain. And then you then know that when N terminus is being talked about, it only means the amine group end. And the C terminus is the carboxylic acid group end. It clearly tells you that when the amino acid is binding with the transfer RNA, it binds with its carboxylic acid end, which is the C terminus. You can't understand if you don't take a jotter and draw and see it. You have to do it. Then you will be able to understand what it is. You read, you take jotter, you draw, and then you see that, okay, this is what it means. This is the N terminus, this is the C terminus. That is the reason why the methionine will always remain at the N terminus. And the last amino acid which will come is going to form the C terminus. All right. So that is an advice. You have to take a jotter and do it. Like I said from the beginning, biochemistry, you have to study it just like mathematics. You can't just be reading it. You have to take jotter and then you try your hands out. The first time, it will be painful if you are not getting something, to be frustrating. But the second time, you begin to get it bit by bit. The third time, you will by all means, get it. So try as much as possible to try your hands over and over again until you have a grasp of these things. Okay, then the termination. So when it reaches the stop codon, instead of a transfer RNA bringing in an amino acid, you see some protein that is coming in there being referred to as a release factor. And what does the protein come in there to do? After it binds to that stop codon, because the stop codon does not code for any amino acid, it will initiate that release factor initiates a process of hydrolysis. It's an addition of water into the complex that will cause a dissociation of the protein, the peptide which is formed. And so there is a dissociation of the whole complex and the protein is released. Then it can go into a process of, if there are some mistakes within the protein molecule itself, it will undergo processing where the portions that are not needed are removed out of it, and the whole protein is now a functional protein as an enzyme, as a neurotransmitter, as a hormone, or whichever protein, whatever function that it has, it goes and starts to perform such a function. So this brings us to the end of today's lecture on nucleic acids. Is a second part of nucleic acids as we started previously. All right, so now the floor is opened. If you have any questions, any contributions, you can raise up your hand. And then, okay, so the first person is Ofe. So, Ofe, you can unmute yourself and speak. Oh, Ofe has put his hand down. He doesn't want to talk again. So anything anybody wants to say, any contributions, any questions, just put up your hand, and then I will allow you you will unmute your mic and then you speak. Yes, somebody's hand. The Kuma Elizabeth. Yes, Elizabeth, you can speak now. Elizabeth, unmute your mic and speak. Oh, Elizabeth
please, I wanted to... Hello, sir.
Yes, we can hear you.
Sir, please, I wanted to ask that after the translation and the question is being formed, like where does it undergo folding?
Okay, so all the process we are talking about is occurring in the cytoplasm. So the folding will be occurring in the cytoplasm. The only thing which occurred in the nucleus was the transcription, the copying of the code onto the messenger RNA. Once that is done, the messenger RNA moves into the cytoplasm. And in the cytoplasm, you find all of that processing being done. Of course, if you want to state specifically where it occurs, then now you go talking about the specific organelles that are responsible for it. So you go for the Golgi apparatus, the endoplasmic reticulum, which play different functions in that process. Is that okay, Elizabeth?
Okay, thank you.
You're welcome.
Yes.
Okay, so who is speaking? Joseph? Joseph, you can unmute yourself and speak.
Hello, sir.
Yes, good evening.
Good evening, sir.
Good evening. Joseph, we are listening. You can speak.
I want some questions. Yeah, okay.
Joseph, we can't hear
Break
you.
down into.
Joseph,
Okay, I'm asking that. Please, can you hear me, sir?
Yes, we can hear you. I can hear you.
Okay. Okay. So I was asking that in life, it looks like we are taking in amino acids, as in the protein. We take the food, cook it, break it down, and then at the end of the day, the body tries to rebuild. The lecture that we had today, that's what I saw. And also,
okay, I didn't hear some part of what you said. The somewhere from the...
Listen.
I didn't hear some part of what you said about the proteins you take in. The proteins are cooked, they break down, and then from there I didn't hear.
Yeah, okay, they break down to their amino acids, primary structure. The body tries to. So with this process, along the way, if someone lacks some of these essential amino acids and it is not corrected, it may lead to some. Okay, so how can we try to maybe educate people so that they know a sense of these things that we are learning? Because we have the privilege of learning them, but others may not have the chance to come across these things that we are learning. And the second part of my question is, I did not see topoisomerase in the reactions that we saw during the lecture.
Okay, so yes, the first part of what you said, Yes. When you take in food that is having some proteins in it, as you rightly said, you cook the protein, and so when you cook the protein, you have actually denatured it in a way. So, but if you denature the protein, it doesn't mean that the protein has broken down into amino acids. No. What the only thing you have done is that you have destroyed the, say, secondary structure, for instance. But the protein still remains as it is. But once you destroy secondary structure, tertiary structure of the protein, obviously the protein will no longer be functional. Fine. Now you have a denatured protein that enters into your body. And that denatured protein is still having amino acids in it. And as it goes into the body, the digestive system now breaks down that protein into the amino acids that you rightly said. And of course, after that process of breaking down into amino acids, then that amino acids will now go into the synthesis of the proteins that your body needs. Because, for instance, if you take in proteins from plants or you take in proteins from some other animals, some animal food, already they are denatured, so they could not be active in any way. Then, of course, the specific proteins which you need to function in your body, as say enzymes or neurotransmitters or hormones or whatever, they are not the same as the proteins which you have consumed in food. So, of course, the body will have to build its own proteins, the proteins that it needs for the specific functions. And you learned from first year, some of these amino acids are essential amino acids. Some of them are non-essential. The essential ones are the ones that the body cannot synthesize. Some of them, the body, can synthesize. And so they are being referred to as non-essential. Of course, there is no thin line between these amino acids. Some of them, some books will tell you is essential. Another book will tell you non-essential. So some of them are not really kind of, it's not a very distinct line between non-essential, but you can have the idea that yes, there are some amino acids that the body can synthesize, some they cannot synthesize. In any case, the body needs a balanced diet. So the simple thing you can tell anyone, so the part of the question we're asking, that you have a privilege of studying this thing, and how can you let others also understand? Simply, it's about taking in balanced diet. It's not only about proteins. Everything, the food you take should have, a combination of different nutrients in it to cater for all the needs of the body. Now, the second part, you were talking about topoisomerase. Yes, the function of the how do I say, the process of gene expression, as was being presented here, really did not include processing. Like, after the protein is being synthesized, we didn't go ahead to be looking at things like processing of the protein itself. We only looked at, the general process of, that central dogma of molecular biology, of having the code from DNA copied onto messenger RNA and then to the protein. Of course, this lecture is a short lecture that you can't expect to talk about every single thing of like, say, maybe cell division, if you go into process of mitosis, meiosis, and all those things. You can't talk about all of them in one. And if it's even not in how do I say, the course outline itself or your syllabus to talk about these things. But fine, if you are interested in some of these things, you can read further and further. You find so many information, a lot, that you can't cover it. Yes, any other question? I've seen that some people are tired and they are already leaving. So if you have any, coming, Matthew. Matthew, you can unmute yourself and speak.
Okay. So please, I want to know why is it that the stop codons do not code for any amino acid?
Okay, so the question of why I cannot answer.
Okay.
We only know that they don't code for any amino acid, and because of that, they are responsible for the termination of the process of the synthesis of amino acids. Why, I can't tell. I don't think anybody who can answer you on why. We only know that's what it does. That is its function. Maybe if you say why, you say, but because the process must end. And if the process must end, just like you write a sentence, there must be a full stop. So something must stop it. So there is that codon that is responsible for making sure that the process ends. If you get to the end of the gene, how will you have the copied the translated information into a protein? How will you now have the protein, being dissociated from the complex so that the protein now can go into use? There must be something that initiates it. There must be something that does that function. Okay. Yes, call me Matthew. I hope that is okay for you.
Yes.
Okay. I think somebody has written in the chat. So let's see. So somebody says, what is the function of the ribosomal RNA? So, the ribosomal RNA, they actually, in the when we were talking about the structure, I talked about these catalytic RNAs. So RNAs that can act as enzymes. So many of these ribosomal RNAs actually function as catalytic RNAs, like ribozymes, involved in the processing of RNA molecules. They act as some restriction enzymes, going into, say, recycling of the RNA molecules, or if there is the need for some processing of RNA after its synthesis, that some portions of it. So what we saw with how do you call it, the introns and the exons, you have some ribosomal RNAs acting in that process. All right. So that was Ofe Frederick who was asking that question. Yes, it is still coming. You can put your hand down. So So any other? It's now 6:45. So I can see our number is reducing, so which tells me that people are tired. Usually I always try as much as possible not to go beyond 1 hour 30 minutes, because the moment it passes that, you see that people start leaving. All right, let me see. So somebody says, per today's lecture, does it mean the initiation stage of protein synthesis undergoes 3 stages? No. It is not. Okay, I know. Okay, the question is not the initiation stage of the protein synthesis that undergoes 3 stages. I think you want to say the process of protein synthesis undergoes 3 stages: initiation, elongation, and termination. Exactly so. Yes, that's what it means. There will first be the start of the process, which is initiation, and then there will be elongation, and then there will be termination. So yes, you are right about that, as you rightly said. After the initiation process, then there will be addition of more amino acids to give you a peptide, and then as more amino acids are adding, you now, will have a long peptide being formed, and then in the end, you will be having a peptide which then gets dissociated at the end. That is in the process of elongation. All right, so I hope that is clear. Yes, any other? Okay, somebody has written, Richardson. He is saying the topoisomerase is the same as the DNA gyrase, which does the unwind. Oh, okay, fine. All right, thanks for that. Yeah, okay, yeah, yeah, you can say that, because the topoisomerase really, yes, it has that function of unwinding of the structure of, the DNA structure. So like changing, it says topoisomerase, so it's changing the topology of the DNA molecule. So yes, that's true. Yes. You can see, yes. So I mean, you see how the word even tells you clearly what the enzyme does. It says an isomerase, so it's a different structure, but that structure, there is only a difference. If you say an isomer of two chemical substances, some you know that this substance is having different structures, but they are having the same composition. So in this case, as a DNA topoisomerase, the DNA structure is having the same composition. The only thing that has changed is its topology, or let's say it's this time around is not a chemical structure, but it's a topological structure. That's what it means. Yeah, so that's true. Yeah. So Richardson has taken my mind to think much more of the question about the DNA gyrase. All right. Komi's hand is still up. So if there is no other question, no other comments, we have been online for 1 hour 51 minutes. So I want to move the motion for the close of our meeting. So if you agree, you can second, so that we end our class for today and look forward to meet the next time. So any secondment for
I
the
second.
proposal? All right, so Elizabeth seconds. Anybody else? If you second, raise your hand. Let's see, so that we know that it's not only Elizabeth that seconds, but there are more people who second them. We know that there is support for us. So AJ, Kusi, Prince, oh, more people. Christian, Moses, Emmanuel, Acoustic. Emmanuel Acoustic, is that your name? Anyway, all right. So then, Grace Aite, fine. So many of us second that we bring our meeting to a close. So see you next time. All the best. Bye-bye.
Bye.